Welcome to this explainer. Today, we're looking at the convergence of biological sciences and computational mathematics. Now, for decades, the fields of biology and computer science have pretty much operated on parallel tracks. Biology deals with the wet, unpredictable, and highly variable nature of living cells, while computer science thrives on rigid logic, data, and algorithms. But that's changing. We're going to explore how artificial intelligence is moving beyond the digital realm and actually altering how we engineer living human tissue.
To understand why this shift is necessary, we have to start with this seemingly impossible question. How do we engineer a beating heart, or a functional liver, or even a severed nerve when we can't fully map the microenvironment it grows in? This is the core problem in regenerative medicine today. It's a biological puzzle so vast that human observation alone can no longer solve it. We need a computational solution.
Okay, let's dive into this. Section 1, the tissue engineering bottleneck. The macro problem we face in medicine is organ failure, and the goal has always been to replace those damaged organs by growing new, functional tissue right in a laboratory. But scientists have hit a significant bottleneck, and that is microenvironment complexity. You see, native tissue isn't just some random pile of cells. It's an incredibly intricate, dynamic, three-dimensional arrangement. You've got cells, structural scaffolds, and biochemical factors all interacting simultaneously. Replicating this physical microenvironment through traditional trial and error lab work is just computationally expensive. So scaling this process to actually grow a full organ requires a completely new approach. Section 2. Algorithms decoding biological complexity. This brings us to a profound shift in our methodology. We're moving from purely physical biology to computational data analysis. Essentially, we are beginning to use machine learning as a specialized translator to decode this biological complexity.
When we talk about machine learning in this context, we aren't talking about magic. We're talking about specific mathematical tools for pattern recognition. Looking at the hierarchy of these tools, we have regression models and support vector machines at the foundational level. Then we move up to clustering and decision trees, and finally reach highly complex deep learning and reinforcement learning models. Each of these serves a distinct, specialized purpose in analyzing how cells behave and how tissues develop.
It's really important to explicitly differentiate these tools. Established statistical models like support vector machines and regression are linear and highly predictable. They are excellent for tasks with clear, structured data. On the other hand, emerging deep learning models, like artificial and convolutional neural networks, are incredibly data-hungry and nonlinear. They are specifically designed to handle the vast, unstructured complexity of biological imagery and genomic data. Section 3, real-world tissue regeneration. Let's move from computational theory into the laboratory to see how these algorithms are actually being used right now. And this brilliantly illustrates a workflow from a recent study analyzing bone tissue regeneration. Instead of a scientist manually trying to estimate how fast a collagen scaffold is degrading under a microscope, an algorithm called a random forest is doing the heavy lifting. It extracts physical features from optical microscopy and translates them into computational probability maps. This simulates cellular growth and provides a highly precise mathematical understanding of a biological process. We see a similar application in cartilage engineering. Evaluating the quality of engineered cartilage traditionally requires an expert to manually grade tissue samples, which, as you can imagine, is a slow and fairly subjective process. Researchers are now testing emerging deep learning models, specifically convolutional neural networks, to automate this histological grading. The model categorizes tiled images of tissue into score groups with high accuracy, which completely standardizes quality control. Section 4, transforming drug discovery. This ability to standardize engineered tissue leads us to a crucial implication: translational medicine. By combining artificial tissue with artificial intelligence, we are fundamentally reshaping the drug discovery pipeline. Think about it. The traditional pharmacology pipeline relies heavily on animal testing, which often fails to accurately predict human responses, or direct human trials, which are slow and inherently risky. But if we can engineer accurate human tissue in a lab and use AI to monitor it, how does that change the way we test the safety and efficacy of new medications? Now, what's really interesting about this slide is how established research approached this. Scientists created tiny strips of human cardiac tissue derived from stem cells.
They applied different known drugs to these beating tissue strips and used a support vector machine to read the precise changes in the contractile force. The machine learned the boundaries of different drug classes. Then, when introduced to a completely unknown compound, the FVM could autonomously predict its mechanistic effect just by observing the engineered tissue's physical reaction. The implications for patient safety here are profound. In fact, a data-driven machine learning approach has already demonstrated the ability to predict tissue-specific adverse events, such as drug-induced liver injury or unexpected heart attacks, with an average accuracy of 72%. By testing on engineered human tissue monitored by AI, we can identify toxic compounds long before they ever reach a human patient. It makes drug development faster, cheaper, and significantly safer. Looking a bit further ahead, we reach a speculative horizon. While we currently use AI to analyze tissue, the next theoretical step is using reinforcement learning agents to actively grow it. In this scenario, an algorithm connected to a bioreactor would continuously monitor a growing tissue's metabolites and structural properties. It would autonomously adjust the electrical stimulation or nutrient flow in real time to optimize its growth. The environment drives the algorithm through a reward system, which could eventually automate the most delicate aspects of biological engineering.
Section 5. The interpretability trade-off. Integrating these algorithms into critical medical decisions introduces a unique set of intellectual and clinical dilemmas. We have to weigh the tension between an algorithm's raw performance and the clinical necessity for transparency.
This is known in the field as the interpretability trade-off. Deep learning models can achieve incredibly high predictive accuracy when analyzing medical data, but they operate as a sort of black box. Even the programmers often can't explain exactly why the neural network arrived at a specific conclusion. Conversely, simpler models like decision trees offer high clinical transparency, meaning you can follow their exact logic step by step, but they sacrifice some predictive power. In medicine, where doctors absolutely must justify their clinical rationales, relying on an opaque algorithm, no matter how accurate it is, remains a significant ethical and practical hurdle.
Section 6. What biology becomes next. As we step back to look at the macro impact, it's clear that this convergence is the foundation of true precision medicine. We are moving toward a future where therapies and engineered tissues are highly personalized, based entirely on a patient's unique genetic and biological data. But to actually get there, we must overcome the data bottleneck. The efficacy of AI is entirely dependent on the quality of its training data. Right now, the field struggles with isolated data silos, variations in formatting across different labs, and restrictive privacy policies regarding medical information. Furthermore, living cells do not have fixed chemical compositions. They are inherently variable. Standardizing biological data so that machine learning models can accurately interpret it on a global scale is the next necessary breakthrough. So, the crucial point is that the integration of artificial intelligence is what allows tissue engineering to scale. It provides the necessary tools to predict scaffold performance, analyze cellular responses, and eventually synthesize patient-specific organs and test personalized medications. We are transitioning from an era where biology was purely physical observation into an era where biology is analyzed as a highly complex data set, and it leaves us with a compelling thought to ponder. When the intricate behaviors of human cells become fully translatable into computational data, what are the limits of what we can actually engineer? If you're interested in where science is heading, like this video and subscribe to Beyond Biology Lab. Press the bell icon to stay updated, where science meets tomorrow.